Hello friends, before starting the topic today, I would like to ask you, have you ever heard about the human insulin hormone which fights the diabetes? About the hepatitis B vaccine which is made from yeast cells? Or about the resistant plants such as the Bt cotton, which is a genetically modified organism that fights against various pests? Have you not wondered how scientists are able to achieve all this? Today, in this video, I will be discussing about a very powerful technique, that is recombinant DNA technology, which has helped man overcome diseases and improve his environment. So first, let's look into what is recombinant DNA technology. It is a technology by which a recombinant DNA molecule is produced by joining together two or more DNA segments, usually originating from different organisms. It enables the DNA from different sources to be identified, isolated, and recombined so that a new characteristic can be introduced into an organism. A recombinant molecule has a coding region from one organism joined to a regulatory sequence from another organism. For example, to produce the human insulin hormone, we will need the gene that codes for insulin in human, along with the regulatory sequences from another organism, and this recombinant molecule will be inserted into a bacterium so that the recombinant bacteria gets a new characteristic that is to code for human insulin hormone. Now, what is the objective of this technology? The objective is to obtain a large number of copies of specific DNA fragments and to recover a large quantities of the proteins produced by the concerned gene. For example, like insulin hormone, vaccines, etc. As this technique aims in producing identical copies of the concerned gene, it is also known as gene cloning or genetic engineering. Now let's look into the steps that are involved in DNA cloning. The first step is isolation of the DNA, then recombinant DNA formation, and then transferring it into a host, that is host transformation, and then selection of the transformed colonies. Firstly, let's look into the first step, that is isolation of DNA. When we isolate the DNA from a cell, we get the whole genome of the cell. Now, how do we get our desired gene of interest? For that, a special molecular tool called the molecular scissors are needed. Now, what are molecular scissors? They are a group of enzymes which can cut the DNA at specific sites, and thus the gene of our interest can be isolated. We also need to select a carrier molecule which can self-replicate, and such DNA molecules are called vectors. An example of a vector is a plasmid that is present in a bacterium. Plasmids are extra chromosomal self-replicating DNA molecules. Now, firstly, we need to prepare the gene of interest and plasmids by cutting them into with the help of restriction endonuclease. Once our gene of interest and the vector is ready, the next step is recombinant DNA formation, in which the gene of interest and the vector is joined to form a single self-replicating DNA molecule, and this is known as the recombinant DNA. The enzyme DNA ligase covalently ligates the two DNA segments and joins them. The recombinant DNA molecule comprising of the vector and gene of interest, and the vector that is to be used as a carrier molecule has to have few components that are very important. The first component is the presence of orisite. The orisite gives the vector the ability to self-replicate in those cells. Secondly, a unique restriction enzyme site is needed, as the vector has to be cut open to insert the gene of interest. And last, a genetic marker which will help in the selection of host cells containing the recombinant DNA molecule. After formation of the recombinant DNA molecule, the next step is host transformation. Now the recombinant DNA molecules has to be transferred into a host cell, and it can undergo DNA replication independently of the host cell chromosome. And at the end result, it will amplify the gene of interest inside the host cells. Now there are two methods of host cell transformation. The first method is attack strategies, in which it includes the techniques to introduce the foreign DNA directly into cells. For example, the biolistic and microinjection techniques. And the second technique is a strict slit strategy, and this includes the techniques to introduce DNA by persuading cells to take up DNA from their surroundings. In this technique, like the calcium phosphate precipitation, liposome mediated transfection are the common ones. We will study in details about all these methods later. For now, let's proceed to the fourth and last step, that is selection of transformed colonies. The transformation process are not 100% efficient. So it leaves a combination of host cells carrying the recombinant DNA molecule and those that do not take up the recombinant molecule. Thus, the host cell, those are transformed, must be selected. It requires the presence of suitable marker genes. As I have already mentioned, the requirement of a marker gene in a vector is very important, because the expression of the genetic marker will provide a means of identification of host cell transformation with the gene of interest. One of the commonly used genetic marker is the antibiotic resistant genes. And antibiotic resistant genes, after the transformation of the host cells, now to select the host cells after the transformation, they are grown in an agar plate containing the antibiotic against which the vector has the antibiotic resistant gene. Thus, the host cells which are transformed with the vector will grow in the antibiotic plate as they contain the antibiotic resistant gene and can be selected. Now, the clones that are transformed will be allowed to grow for gene multiplication, expression of proteins as per the requirement. In this video, I briefly summarized the recombinant DNA technology steps, but each step requires various molecular tools that we will study in our next video. If you have liked my video, do click the like button and press the bell icon for further upcoming videos. Thank you. Bye-bye. Hello friends. Before starting